Today, our lecture is based on histology of liver. Histology means study of the tissues. So, liver is the largest organ in our body. It is a gland present in the abdominal region. Embryologically, it is derived from the endodermal layer. It is involved in several important metabolic activities such as digestion, synthesis, storage, excretion, and so on. However, its diverse functions are not reflected by its component parenchymal cells, which present a uniform structure. Liver, it is a reddish brown in color. In rat, it has 4 lobes, and in dog, its lobe is 7. In man, there are 2 main and 2 smaller lobes. The posterior surface of the liver at right lobe shows a short, deep transverse fissure called porta hepatis or hilum. The liver is completely covered with fibroelastic connective tissue forming the capsule of Gillies, outside of which is an incomplete serosa. At the porta, the capsular connective tissue passes into the substance of the liver and divides it into internal framework. This connective tissue stand also and support the blood and lymph vessels, bile duct and nerve which pass through the porta hepatis and permeate the entire liver parenchyma. In the parenchyma, the connective tissue enters and make lobes. The hepatic veins and their tributaries course separately through the substance of the liver, and very little connective tissue is associated with them. Due to the connective tissue, the parenchymal substance of the liver is divided into compartments. These compartments are polygonal in shape and called hepatic globules. So, hepatic globules are the structural and functional unit of the liver, measures about 1 millimeter wide and 2 millimeter long. In section, the lobules appear polygonal and are closely opposed to each other. In some animals like pig and camel, their polygonal nature is seen clearly because of distinct connective tissue. They are also called Gillies capsule. But in man and most of the other mammals, the capsule is restricted largely to places where 3 or more lobules meet. Every such area where the lobules meet to each other is characterized by a branch each of the hepatic artery, portal vein, lymphatic channel, and bile duct. This is portal area. That means in between the many tubules, a space occurs. This space is called hepatic portal area. In this hepatic portal area, presence of the hepatic artery, hepatic or portal vein, and lymphatic channel and bile duct is present. On the basis of these, the artery, veins, and bile ducts are known as commonly hepatic artery, hepatic vein, and hepatic bile ducts. At the center of each lobule, presence of a central vein is there on which converge radiating rows of liver cells or hepatocytes alternating with blood spaces called hepatic sinusoids. That means from the central position, central vein is present from which the row of hepatic cells arises in the form of the cords. These cords are called hepatic cords. In between the two cords, presence of space is there. This space is called, this space is filled with the blood and known as hepatic sinusoid. Blood enters the liver sinusoids from small interlobular arteries and venous branches flow through the lobules and leaves the central vein. As it passes through the lobule, the central vein receives the sinusoids from all sides and increasing in size leaves the lobule at its base. Here, it unites with the central veins of other lobules to form intercalated vein. Several of these open into the collecting vein and then in turn drain into the hepatic vein. So hepatic cells or hepatic cords itself separated to each other by the help of hepatic sinusoids, which is clearly seen into the figure. Hepatic sinusoids, as you know, it is nothing but the space which is made by the intralobular system of blood capillaries which convey blood from the portal areas to the central vein. They are wider than capillaries and anastomose irregularly. The sinusoids separate the hepatic lamellae from one another. Their lining cells are in direct contact with the hepatocytes, there being no intervening connective tissue occur. It was believed for a long time that the lining cells of sinusoids are of two types: a thin endothelial cell with dark and compact nucleus and sparse cytoplasm, and large phagocytic Kupffer cells with a bigger oval nucleus and branching pseudopodial process. It was also accepted that if needed, the endothelial cells could transform into the Kupffer cells. However, electron microscopy and histochemical techniques have very recently established that there are three types of cells population having different origin and functions. These are the sinusoidal endothelial cells, Kupffer cells, and perisinusoidal fat storing cells. The modern view about these cells are as the Kupffer cells are large, irregular, amoeboid, phagocytic, macrophage cells derived from monocytes. They belong to the reticuloendothelial system. They are not fixed in one place. Kupffer cells are situated on the endothelium with thin process extending between the endothelial cells. Their ovar nucleus often shows an identification. The plasma membrane exhibits numerous folds, villi, and sinusoid invaginations. The cytoplasm is enriched with the usual organelles. It frequently contains engulfed, worn out erythrocytes, pigment deposits, and granules rich in iron. New Kupffer cells are produced from pre-existing ones. So, such important Kupffer cells are present in the liver, which is helpful phagocytic in function. So, besides this, the hepatic cells are also present in the liver, which is helpful for the detoxification and digestion phenomena of the liver.